So much, sir, for enlightening the importance of organochips and AI/ML importance in drug discovery and development. It really added essence to your presentation. Now I request all the students and delegates who have presentations please go to the pharmacy campus and start your presentations. Oral presentations. Only oral presentations, students.
Not all.
He earlier worked as PK expert and head of pharmacokinetics, biostatistics and medical writing department in MNC companies like Novartis and Clinisync Clinical CRO. Dr. Jagan has completed his B.Pharm, M.Pharm and PhD from Kakatiya University. He completed his postdoc from Florida A&M University, USA in nano drug deliveries in cancer models. More than 25 papers and 50 presentations at national and international level and 2 books are in his credit. He also received UGC Fellowship in Masters and AICTE sponsored National Doctoral Fellowship during his PhD. He received several travel grants to travel overseas to present his scientific works. He also completed his PG diploma in patents law from National Academy of Legal Studies and Research. Ladies and gentlemen, let's invite Jagan sir with a good round of applause. I request you, sir, please deliver the presentation.
Thank you. Thank you for such a nice introduction. And once again, my thanks to the Malla Reddy College Management, Principal Sir, Sudhakar Sir Garu, and Coordinator, and Surya, your teacher, your beloved teacher, for inviting me to here. Very good morning, everyone. Okay, super. So, let me start the session with a question. First of all, I should appreciate you because today is second Saturday, right? It's a holiday to most of the colleges. In spite of that, early morning, turning out in such a huge strength, a big round of applause to all of you for your enthusiasm, spirit and energy. I really appreciate. Okay, thank you. So, but I have seen a little disappointment when I said let me start with a question. Are college में भी questions पूछते teachers and guest speaker भी questions पूछ रहा है. But I will ask only one question. So who would like to answer that question? Very simple question. In fact, I will support if you have any problem. Means I will give the hint. Who would like to take the opportunity? Do we have the other mic? It's okay. Whoever wants to take this opportunity can come on to the dais. I will give my mic. And I will support you in case if you face any difficulty to answer. Who wants to grab this opportunity? Coming on to the dais in Malla Reddy Sir College campus in auditorium. Great. Welcome. Yeah, please come on. You want water? Give him some water bottle. Who wants to take the opportunity? Simple question. Yeah, very good. Great, madam. Super. Please, big round of applause. The question is, are you ready? Yeah, very good morning. Are you feeling tense? No, right? If you wanted to have some water, you can take water. You are cool, right? It's not the KBC, not Kaun Banega Karodpati. Okay, right? So the question is, my dear intelligent pharma students, the question is, what is the answer for 2 plus 3? I repeat the question. What is the answer for 2 plus 3?
It's obviously 5.
Exactly. Please, big round of applause once again. Candy, can you please bring the chocolate? You can come from that side. Yeah. Please take this chocolate. Please, the photo. Thank you. Congratulations. Yeah, you can leave the stage. See, please be here for a while. See, the big round of applause is not for her aptitude, but for her attitude of facing anything, no matter what's going to come on the stage. That's the attitude you have to build up, my dear intelligent friends. This is the message I wanted to give this means in the Malla Reddy campus today. main motive of my arrival to here. My job is done. What I am going to tell you now, the bonus. जो भी बोलता हूँ ना अभी, bonus. My job is done. Build that attitude of facing anything in the outside world. You can reach any point. Because most of the times what happens, when you get into the real world, more than your aptitude, your attitude decides your altitude. So please take this message lifelong. जिंदगी भर याद रखना है, मेरे को नहीं, मेरा words को. Thank you, madam. Thank you so much. Yeah. So, going back to the next slide, please. Are you able to see the slides clearly? Okay. Are you able to hear me loud enough? Okay. That's the energy, because we should feel it like, the pre-release event of Pawan Kalyan movie. Okay, right? So that's the energy you have to show throughout the session. Those sessions will happen in the evening and this is happening in the daytime. That is only the difference. The rest of the energy, enthusiasm should not change. Same to same, okay? So, why did the rest of the people, means I encourage, in fact, if she was not able to answer, means I wanted to even support her like 1, 2, 3, 4, 5, something like this. I told you, the question is going to be very simple. In spite of that, from the auditorium, nobody had the guts to come onto the dais, right? The same thing will happen in the real world also. Most of the times what happens, the actual challenges, actual problems are going to be as simple as this. The size of the problem is going to be as little as this you have witnessed on the stage now. Unnecessarily, you assume it to be the big. And you assume it to be such a big size, you don't even dare to come onto the dais, you don't dare even to attempt the problem. That's where we lose the opportunities. That's where we lose the opportunities. No matter what, keep attending all the interviews for all the high-fi opportunities. Definitely, because this is what even in the real world scenario also happens. You don't even attend where you are eligible, where you are deserved for. So you should not lose any game, not by even participating. That should not happen, my dear friends. Okay? So you should not have that fear of failure. In our current generation, I have two news. One good news, bad news. Which one I should tell first? Good news or bad news? Okay, bad thing I should tell first, right? I don't want to disappoint, but it's okay because you asked me to tell the bad thing first. Okay, I will tell. The EQ of the current generation, means your generation, is very, very bad and very, very poor. Means you don't have the habit of accepting the failure. You have least tolerance for the failures, to accept the failures, which is not the case in our time. Okay, right? We used to take, जो भी होता है, होने दो, it's okay. Light लेते थे हम. But it's not in our case. Even you are taking the smaller things deep into, personally. Okay, right? So that's the bad part in your generation. Please try to change. The good thing is, your IQ is much, much, much higher than us. If given a course, 7 days course, if a new course comes, Dr. Chandu rightly said, AIML, Artificial Intelligence and Machine Technology, is the current trend. If a course comes, 7 days course comes, for example, if I lose my job, and if a new course comes, if we all attend, means if we all go to Amirpet and if we all take a course, including your Surya sir and Dr. Chandu and myself, probably you will learn faster, much, much faster than us. Because your IQ, your intelligence quotient is much higher than us. You can learn anything so fastly and you can crack any interview also. But, after going into the job, I see many people, they are leaving the jobs because of some inferiority. Means they have a very good IQ because they coming from some middle class or below middle class or from rural background, they are unable to adjust to the corporate culture. it's a cultural shock. They unnecessarily assume about themselves that they are inferior to somebody else, which should not happen. Even because of that, people are leaving the jobs. Okay, right? So that should not happen. Your IQ levels are very high. And see, in the pharma sector, he said, Dr. Chandu said, we are hardly producing any new molecules, NCEs, right? We have become just like copycats. That's what the generic drug industry. We are copying somebody else. We have almost 130, more than 100 crore population, right? India में intelligence के कमी है. No, right? We have so many intelligent people, right? Even then, why are we not producing new NCEs? Why are we simply copycatting? Simply we are making the generics? Why our most of the focus is going on to the generics? Because we are a middle class family. Middle class in sense, I'm talking about India. We are above middle class or middle class. Because already whatever the medicines are there, we are not able to produce and we are not able to make it in affordable cost to everyone, right? And where do we have the room to think of a new NCE, right? It's a matter of money. NCE, see, please understand again, even Dr. Chandu also said the same. It's a matter of money, not the intelligence. NCE is just not about the intelligence. It needs huge amount of the money and we don't have such huge amount of the money in the parliament or in the assemblies to allocate for the research. And no private company will come forward to invest or to gamble such a huge amount of money where the success rate is very, very little. Out of 10,000 to 20,000 molecule, one will see the light. Means what is the success rate? It's very, very little, right? How risky it is. Again, I'm telling you whatever I am telling on this dais are purely my personal opinion. Okay? Strong disclaimer. Nothing you can attribute to my organization. Okay, right? So no company is running the charity. No company is running the charity, right? They have to make money out of that. Otherwise, they will get into the losses and everybody will lose the job. So, that's the reason. So, please understand positively that India is not producing new NCEs means it's not because of the intelligence lack, but because of the money gap. Okay, right? It's a matter of money. So, again, coming back to, please never lose any opportunity because of the fear of failure or fear of criticism, okay, right? Which is a drawback of your generation, okay, right? Anedo means if I ask a different question, if she unable to answer, it's okay. What's wrong in that? At least she had the enough guts to come onto the dais, right? Who means receives in the Telangana more criticism? Can you please name two people who receive every day in all the channels, who receive the highest level of criticism? Either KCR or Revant Reddy, right? Who are the most successful people? Who are the great leaders today, as and today? Is it ever? So, does anybody in the morning in the TV9 or TV5 debate about you and your merits and demerits? Nobody will care. Whoever get the criticism means they are in a very good position. So, please have the guts to accept the criticism. Next slide, please. So, the fear means, see, false evidence appearing as real. There is nothing actually there. You imagine yourself, it's an illusion. Next slide, please. So, when you get a fear, you have two options. Okay? What is that? Forget everything and lose the opportunity and run away from the opportunity, or face everything and take the opportunity. Next slide, please. So, that's in your hand. Next slide, please. Next slide. So, coming back to our technical session. So, you are all boosted enough, right? You are all with me. Yes or no? Thank you. So, innovation in generic clinical trials. So, have you ever heard in the past about the generic clinical trials? At least generic medicine? Please raise your hands, whoever already know this term, generic medicine. You all know, right? What is the major difference? Next slide, please. Okay. Yeah, please, next. So, our show items, these are the index menu of my presentation in the next 30 to 40 minutes. Myths about the generic drugs, the cost secret of the generic drugs, and introduction, basic BE concept, what is meant by super generics, and how we should develop out-of-box thinking, even to make innovation in the generics. And the BE concept through the terminology, and the last one, if I get time, if we still have the time, I don't think, if we still have the time, we will have a second show. We will have a surprising second show. I will means एक ही ticket, 2 movies. First technical, then means what you like most. Okay, right? Next slide, please. अच्छा है ना? Means you are liking the session. Okay, right. Next slide, please. That was the innovation. Means we can take the opportunity to inculcate, to implement our innovation everywhere we want. There is no such place where we cannot make innovation, right? In everywhere means the way you write, the way you search for your opportunities, everywhere you have to go in an innovative way. Then only people will see you. Then only you will get your own identification. Is it or not? So, what are the myths? Generics contain relatively less drug. When I go to my village, they also say, can you please take the generic drugs? Means मैं सुना हूँ उसमें आधा ही रहेगा drug. Means only they keep less amount of the drug. क्योंकि 50% discount देते ना. Means they give 50% discount. Means they keep only 50% medicine. That's what the impression in the common people. Being pharmacists, being the scientific community from the pharma, it's your responsibility, my dear intelligent friends, to bring that awareness. In parallel, while pursuing your own education, it's also your responsibility. Start from your family. Raise the awareness. Generics don't have other drug. They also have full drug. You can take generic drugs as well. But price to आधा ही रहेगा और सस्ता भी रहेगा. If a tablet costs, especially in the oncology, or injection costs 2 to 3 lakhs, the generic version costs 10,000 to 15,000. If it comes to the tablets, even a 10,000 rupees will cost tablet will actually in the generics, it will cost a maximum of 80 rupees to 100 rupees. Such a huge difference. So, next, generics are not as safe. These are all the myths. Have you ever heard these myths? Do you also have these in your brain? We will be getting the answer in the next slide. Okay? Generics are not effective. वो काम करता ही नहीं. Generics take longer time to act in the body when compared to the branded or innovators. Or generics are made in substandard premises so that they can make it anything and they will just throw it. That's why they are coming at the cheaper cost. Next. But what's the truth? Then means if everything is same, branded to generic, if everything is same amount of the drug, same quality, same premises, then why do they offer at a very cheaper cost? Innovators are mad people. So this is the question generally that revolves in the brains of the common people. Okay, right? Yeah, next please. So generics and the branded both are safe, effective, and the quality. Everything is same. Only the difference is generics are affordable and innovator or branded medicine, innovator medicine is not affordable. The reason is, next slide please. Yeah, next slide. US healthcare system has saved 408 billions in 2022 because they released this report last September, just few months back. So I read the report. What I realized is that they saved 408 billion and we are hesitating being in India and what we feel like the most advanced country, the US people are using the generics. Okay, right? Next slide please. past 10 years, approximately they saved the 3 trillion US dollars. 1 trillion means how many rupees? Can anybody answer? 1 trillion, at least how many zeros? At least 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 12. Very good. 12 zeros. After 1, there are 12 zeros. Means you cannot even calculate, you cannot even type in your mobile calculator. Right? So, 1 trillion means 12 zeros means how many rupees? Means at least how many crores? 1 trillion means 1 lakh crores. 1 trillion US dollars means 1 lakh crores. Right? And 83 rupees, right? Means at least 80 rupees. 80 lakh crore rupees in Indian currency. 3 trillion means 80 into 3, 240 lakhs crores they saved. What is state budget? 1 lakh, 2 lakhs. See, in the just last 1 year, 408 billion. Billion means how many crores? 100 crores. 1 billion means 100 crores. We all hate mathematics, and again you are teaching mathematics. Is that what you are thinking? Even I also scared about the mathematics. That's why I took a BPC, right? So, 1 billion means 100 crores. That is in dollars. In Indian currency, 1 billion means, आज के हिसाब से 8,000 crores. 1 billion means 8,000 crores. 408 billion means 40 lakhs crores just in 1 year saving. I'm proud to say that this saving happened majorly 80% because of India. This saving happened for the America because of India. Because India is the major exporter for the generics. Okay, right? So, if you put the budget of all the states in India together also, it is lesser than that. 1 year. So that's the wonders they are doing. So when they are doing, see, the literacy rate is almost 100%, right? From a long back. So that everybody understands what is fact. That's why they are taking the generic medicines in US. So please do create awareness. Okay, next slide, please. Next. Yeah. Nearly 9 out of 10 prescription drugs filled in the US today are generic drugs. For every 9 drugs in a prescription, doctor writes out of 10, 9 generics. Means out of 100, 90 and more are generic only. But the surprising thing is that, next please, yeah. Though 90% of the medicine on every patient prescription is generic, they contribute only to the 20% of the total expenditure. Okay, right? These 90% of the medicines contribute only 20% for his bill from his packet. Only those 10% innovator medicines are eating the 90% of his money. Though the statements are a bit confusing, hope you got it right. So, only because of the lack of the awareness. So this is important, means considering the society perspective, your responsibility perspective. Okay, right? Next slide, please. So why this cost difference is coming? Just keep pressing. Yeah. Please, yeah. Continuously, yeah. See, when you look at the innovator drug, they have to do some different studies, right? So these are all the studies. Okay. 1, 2, 3, 4, 5, 6, 7. CMC, we call it CMC, Chemistry, Manufacturing, Controls and Labeling, in vitro testing before entering into the market. They are common for both generic and the innovator. But where the most of the drug will go right? To see one new chemical entity the light, if you want to bring one new medicine into the light, on and average cost it is 2 to 3 billion. Means it range from 2 to 6 also depending upon the molecule that you choose, but on an average at least 3 billion you can say. Or as rightly said by Dr. Chandu, let us take only 2. Okay, right? Even then what is 1 billion US dollars means 8,000 crores. 2 billion means even in a worst case scenario, we need 16,000 crores to bring one new molecule into the market. So where we invest this money means majority of the amount will go into the 6 and 7. Preclinical tax, means whatever you say. Preclinical and clinical studies. Right? And we don't need to do all those studies. Because innovator already did all these studies. And he said that this is the molecule, works super in so and so cancer and is very safe, established. Okay? For that he invested some 20,000 crores and he brought a new molecule. What I will do? Sir, I'm exactly same to same this person, no difference. Please give me approval. That's it. I do get the approval. So now how will it make a difference in taking A or B when both are same? Everything is same. Means whatever the API, active pharmaceutical ingredient, tablet में जो रहता है API plus excipients, right? Everything is same. Whatever API he uses, the same API I will use. If he uses a diluent, I will use the same diluent, HPMC. If he is a binder, I will use the same binder. So, for example, if he is SSG as the integrant, I say sodium stearate as the lubricant, same. Talc as the gradient, same. Everything, if total filler volume 900 mg, 900 mg, उसमें drug 100 mg, 100 mg, tablet shape oval. I will keep everything same. When there is no difference, so why shouldn't we take the generic? If that is the case, there is a small problem that why should people, innovator people invest too much of the money? That question may come in our brain, right? So, the patent office also thought the same. Means innovation should not be discouraged. For example, there is a canal and people are going all the way around to reach the other side of the canal, which there is also a village. They are going 50 kilometers away, but somebody came forward to construct a bridge. Right? For him to get his money back, he will be charging some toll, right? 100 rupees per vehicle. If he collects for 10 years, he will get his money back, right? The same will be done. Here we will give the royalty, means the patent period. We will give some patent period. Okay? During that period, he only will sell his product. Nobody should come into the market. Once the patent expires, anybody can make the generic version of that. Nothing wrong in that. Because innovator already got his money back. Right? Innovator already got his money back. Okay? Even after the patent expiry also, listen to me very carefully, even after the patent expiry also, if nobody makes a generic version of that, what will happen? Innovator will continue to sell his medicine for the same price he started with. The beginning price is to get his money back within a span of 10 years. That's why he put a large cost. But if nobody cracks, if nobody makes the reverse engineering to crack what exactly the molecule is, how exactly it has, because not always all the medicines are straightforward medicines. So much of research goes into even to make a generic. It takes so much of the time, right? For example, tomorrow Sunday, right? I am a non-veg, honestly telling. Most of you might be the non-vegetarians. Okay? So whoever eats non-veg, if I give you same ingredients, okay, I will give to two people 1 kg chicken, 1 kg chicken, 1 bundle of curry leaves, 1 bundle of kotimira and pudina and same spices, onion, do mirchi. Will they make the chicken curry with the same taste? Obviously not, right? The ingredients are same. So, here the taste, smell, quality, all these are the evaluation parameters. In the similar fashion, even for any generic medicine also, there is a evaluation parameter called BE, bioequivalence. For your chicken curry, taste is the BE. If both are same taste as that of the Zomato, yes, you are bioequivalent to the Zomato. Same taste aa gaya. But in my case, if I bring the same systemic exposure, I say, have you already come across in your textbook bioequivalence? What is the two key terms to define the BE? Rate and extent. Rate and extent. These are the two key terms that define the bioequivalence. So, for example, if I take the generic, means innovator medicine, whatever the tablet, means what will happen? I will take the tablet followed by glass of water so that first it will undergo disintegration, right? Disintegration means losing the integrity. Means the tablet will become small pieces, right? Granules. Initially we take the granules and we will compress and we will make the tablet, and the tablet again will become the granules in the stomach. And then what will happen followed by disintegration? Dissolution. Means conversion of the granules into the solution state. Because granules as such will not make the holes to my stomach and they don't get into the blood, right? Anything to get absorbed into the systemic circulation, the basic funda is it has to be converted to the solution. Until and unless any dosage form, no matter what is that, the oral dosage form I am speaking about, no matter what is it, whether it is suspension, capsule, tablet, ODT, no matter what, any oral dosage form must get into the solution. That is what called the very critical step, dissolution. उसके बाद क्या होता है? Absorb होता है. उसके बाद systemic exposure आ जाएगा, right? Okay, if that is the same, that is the basic concept. So, in the generic industry, what we are proving is that when innovator medicine is taken by the patient, the same systemic exposure will come by my medicine also. That is what we will prove. This is the basic concept, my dear friends. That will happen through the BE study. That is why I am taking too much of time here. Have you got my concept? Because learning the concept is the most important thing. Okay, right? What's the time? 12:05. Okay, we are running short of the time. It's okay. So that's the basic funda concept. That's what. I'm very privileged. Thank you, sir. Thank you so much, sir. Very nice to see, sir. So that is the basic funda concept. So that is where the cost difference is coming. That is where the cost difference is coming. Okay? Hope you got this. Okay? Next, please. Yeah. Because we are running short of the time. So initially, the background is, long way background is initially, means if the API and excipients are same, means people were under the impression that by default they will produce the same systemic exposure, which is not true based on our past explanation. And we had some bitter experiences in the past. That is what called the phenytoin toxicity, especially for the narrow therapeutic drugs. What happens? Therapeutic window will be very small. So we had a bitter experience. From that, we learnt all the concepts we discussed. Yeah, next please, yeah. Yeah, next. So this is the concept we just had, right? If systemic exposure is same, automatically my assumption that efficacy is going to be the same. So this is the basic funda for all the PK endpoint bioequivalence studies. Yeah, next please, yeah. If any tablet is taken orally, the basic concept is it must get into the solution state, which is called a critical step dissolution. After that, it will get absorbed and it will go into the central compartment and it will reach the receptors in the tissues, and this is assumed to be the same. If same systemic exposure is attained, means we are assuming that the same level of receptor occupancy is going to be there for that particular drug. Yeah, next please. So, when even in the case of where is the innovation coming, probably you might be having a question, but even in the case of a generic also, we have a plenty of the room to bring the innovation, which we call super generics. For example, a super generic is an improved version of the original drug. After expiring the patent, means even we can make a much better version of the innovator also if really we put some innovation into the generic version. For example, Abraxane, paclitaxel drug, Taxol is the brand name. Instead of using the Cremophore, which gives so many allergic reactions and the hypersensitivity, if we use the albumin, it has got much better efficacy and much better safety profile. So it doesn't mean that we cannot bring any newer or the better version of than the innovator. Even generic, if they want, they can make even with the lesser amount of the money also, they can bring, there is always room. So no compartment is closed for the innovation, my dear friends. So in the similar fashion, Candesartan, CVS, cardiovascular drug, means by making a nanoparticle dosage form, we reduced the dose. So most of the AEs are because of the dose, right? Dose dependent toxicity. If you reduce the dose, obviously there is a scope to bring down the toxicity. So that is how we achieved in this case. Next please, yeah. So what I request all of you is that, so develop that out of box thinking to make anything, anywhere, to incorporate some innovation so that you can be differentiated from the conventional, so that you will get a very good recognition. In the pharma also, we always make not only the exactly same to same, sometimes a differentiated product for which we file through a different application. ANDA, ANDA. Innovator is NDA. ANDA means generic version, right? But in between that also, if we make some differentiation thing, that is what called 505(b)(2) application, and for this also you will get some special privilege and recognition from the cost and the identification perspective. See, in the similar fashion here also, just because of the development of the nanoparticle version or the means by improving the solubility, because most of the drugs are suffering from a disease called solubility, which we categorize under a disease category called VCS class 2. Even class 4 also, but the class 2, most of the drugs are the class 2. So for them, if you improve that particular problem called solubility, you can enhance by various pharmaceutical approaches, right? Which you every day read in your textbooks. If you take those deep concepts into your brain and if you apply even to a simple drug, you can do the wonders, right? So the same was done, means when myself and Dr. Chandu were in the Florida, we developed the advanced version, means the novel version, to improve the oral bioavailability. The other thought, out of box thought, again from the Dr. Chandu, he said that if you give some other drug, not at the therapeutic dose, but at the subtherapeutic dose, the telmisartan, even if it is not wanted for the cancer patient, okay, if you give prior to giving the cancer drug, the telmisartan, what it is doing is, most of the cancers are the solid tumors, means they make some compartmentalization because of the collagen. Drug will not get, it is very hard. वो घुस नहीं सकते अंदर. Okay, right? So, in such case, if you expose that particular animal with the telmisartan, we realize that the loosening of the tissue happened, means the collagen disruption happened, so that actual target drug, anti-cancer drug went deeper. So, that is the out-of-box thinking, which we did in our lab in the Florida. Next slide, please. And I will tell you one more. This is also, means there is no boundary for the out-of-box thinking or innovation. For example, generally you make micelles, you make emulsions, okay, right? And you make gel. So what I thought that why shouldn't I combine all this with nano approach? Okay, so I prepared one micelle, I prepared one emulsion, and I reduced their size. When we reduce the size, automatically it is a nano formulation, right? So nano emulsion I prepared, nano micelle. Mi means here, nano means nano formulation. Mi means micelle. Em means emulsion. I prepared micelle in one container, emulsion in another container, okay, which are nano size, and I put them in a gel. That is why I put a name to this drug delivery system called nano mime gel. So here the basic, see whether it works or not, really it doesn't matter. You somewhere have to start with some starting point, right? So see, every nano drug delivery will choose based on its size, charge, okay, or characteristic feature, or the path that it chooses. It will choose a particular path and it will be absorbed. Especially when you apply anything on the skin, the absorption is very, very poor. Is it or not? When you take anything orally, it will get into the systemic circulation, disintegration, dissolution, right? And the solution can easily, because the mucus layer is very sensitive, right? Something can easily get into that. There itself we are facing the challenges for the bioavailability, systemic exposure. However, the stratum corneum, which is a very tough layer, right? It is very hard. But sometimes, for example, if I get the cold, to relieve from that cold symptoms, who should take the tablet? Me only, right? If I get the cold, if I ask you, all of you take the cold tablets, means how does that look? For example, while cutting the vegetables, generally I don't do cooking. So, but if I cut my little finger while cutting the vegetables, okay, right? If something happens, what should I do? If I take the tablet, will that work? To some extent, because tablet doesn't know that I had a cut on my little finger. What will happen? Same sequences, disintegration, dissolution, and all the molecules will get go. And how much drug will come to my little finger? Very little, right? So in such cases, so there are so many things where we need targeted drug, especially in, for example, skin diseases, topical skin cancers, for example, deep melanoma cancer, basal cell carcinoma, where you want locally the drug. No matter how much drug you take, it is very hard to build the concentration in the required place. For that, you have to take a huge amount of the drug, which is practically not feasible. So sometimes we have to develop a topical drug delivery system, but drug is not going. So I told you, every nano drug delivery systems relatively will have a better absorption, and every nano drug delivery, if it chooses a particular path, why should I not combine? Next slide, please. Next slide, yeah. Just keep on pressing, yeah. See, there are two delivery systems if you assume, X delivery system, Y delivery system. Rather than, the concept is, rather than putting the whole amount of the drug in a particular single drug delivery system, why should I not distribute it? Instead of 100 mg putting in the X, if I put X in the A, it will choose the X path and it will be saturated. The rest of the amount will not get into that. If I put in a Y drug delivery system, so probably my assumption is that it will choose Y path and it will choose X path, intercellular, intracellular, something you can assume. And if I combine, if I put X, 50 amount of the drug, 50 mg drug in X delivery, Y amount of the drug in emulsion, micelle, something like that, if I put that nano mime gel, X plus Y system, probably there is a relatively, there is a scope, right? At least it appears. This is a hypothesis. And you do the experiment. So I'm just giving some food thoughts to you, how to think out of box. Not every time we succeed. I already told you, please learn the habit of accepting the failure. Especially in the pharma, success rate is very, very little. We should not get disappointed. We should not get demotivated with the failures. Okay, right? Next slide, please. Next slide. So this is the Conventional approach is one product, one absorption path, one drug delivery system. Why shouldn't we think? Because we don't have the such kind of products in the market. Make some pilot products in the lab as part of your thesis and publish it so that even industry will inspire through your academic innovative research. You can do that. You have the scope because we will be fit into a particular thought process, so rarely we get the opportunity to think out of box because we do have the limitations. Industry people will have so many limitations to think and to work out of box, but you guys will not have such limitations, right? So you can do any hypothesis, make any hypothesis, and you can do the experiment. So please leverage that freedom when you are in the academic zone. Next please, yeah. Next please, yeah. Yeah, next. Next, yeah. Keep on pressing. Concept through terminology. So hope you got almost all the concepts of the BE, bioequivalent studies of the generic. Okay, next please. Yeah. Pharmaceutical equivalence, everything same, means for example tablet versus tablet, capsule versus capsule. Means pharmaceutical equivalent, if you bring a generic, that is called pharmaceutical equivalence. Pharmaceutical alternatives, for example, already tablet is existing, I am making a capsule which is bioequivalent, that is a pharmaceutical alternative. For example, different salt, amlodipine bacillate is there. If I take the maleate, even if you take 18 mg and 17 mg amlodipine bacillate versus maleate, both will have the same 10 mg of the amlodipine. Okay, but they are alternatives. Bioequivalence, I already explained you. Only the two terms that you should remember is rate and extent. Next please, yeah. Next. Yeah, keep on pressing. So, whoever want to get into the generic drug industry, please, my dear friends, you will be getting plenty of the information in your textbooks and in the literature, because we don't need to miss it. Practically, it is also not feasible. But you have to familiarize with these words. If you familiarize with these words, no, cracking interview is not a big deal. I assure you, I will give in writing. I will give you in writing. If you familiarize with these words and the concepts, just these words, if I ask you, if you are able to say what is it, for all the possibilities, okay, right? You have to think of how you should apply out of box thought process. Means, if I do a generic study, should I do in a single center or multi-center? What are the advantages of the single center? What are the advantages of the multi-center? Okay, right? Why, when we go for the multi-center. So you have to put questions for every terminology. Randomize. Why should I randomize? Balanced. Open label. Why should I not do the double blind study? Yeah, next please, yeah. Single dose or multiple dose. Crossover or parallel. Truncate. So, if you familiarize with these words, automatically by default, I will give you in writing also. We will place somewhere. Means by default, nobody needs to recommend you. Okay, right? But you know, especially in case of the basic, means I cannot explain each and every word, but I will tell you. The regulatories most of the times, when it comes to only the generic, they always prefer healthy or patient study. Means healthy. They prefer, even if it is a oncology product, if it is not having a reasonably bad cytotoxicity, means if innovator already demonstrated that it has a very good safety profile in the healthy subject, if we have that evidence, if we have that precedence, even for the oncology products also, we can go with the healthy. Means healthy or means which population we should select, means always healthy, prefer healthy. When it is healthy is not possible, go for the patient based. And single dose or multiple dose. So what is good? Means how should we prove the, because most of the times, even when I was student, I used to get a question in my mind that nobody uses any medicine for any, most of the diseases one time, right? At least we will be taking at least 4 days, 5 days or 6 days, depending upon the treatment regimen that is defined in the approval label. Why should we do the single dose study? What is the use? But you know, when you do the single dose study, then only you can discriminate what exactly the difference between the generic and the innovator. Means if you really want to get very sensitively the actual true difference between generic and innovator, we should prefer the single dose. So in the similar fashion, ask all the possible multiple questions to every word here and get the answer. By default you will get the job. Okay? Yeah, next. Let me check. Mic check. 1, 2, 3. Nobody is sleeping. Thank God. Okay, right. Next slide, please. Sample size. So, in how many patients? Even see, don't make yourself the biostat parts as irrelevant to you. Even that is also inevitably required for pharmaceutical people to apply, especially to define the sample size, which is the most important thing. Okay, right? And so all these things, ask all the possible question. Why should I give this? How little I should give if it is washed out? How maximum I should give? And what is the rationale behind it? Yeah. Next slide, please. Yeah, next. So these are also some terms. Okay, right? Sponsor means whoever do the majority of the studies. Okay, right? And CRO means they are meant for conducting these studies. Contract research organizations. Okay, right? We have IEC, independent ethics committees. They give the approvals. Ethics committees give the approvals for conducting the clinical trials. And you must be knowing, when you do the even animal studies also, you will take some preclinical ethics committee, or for even clinical studies. In the academics also, you should take some ethical approval, ethics committee approvals, academic ethics committee approvals. So in the similar fashion, for every clinical study that industry conducts, we do take the ethics committee approvals, and the regulatory, whoever gives the approval, wherever we would like to launch the product. For example, if it is the US FDA, we should seek the approval from the FDA. If it is for the Europe country, means we should take the respect to geography regulatory agency. For example, EMA. For example, Brazil, ANVISA, something like this. For example, Canada, Health Canada, India, CDSCO, DCJ. So for every country, we will be having some, because the drugs are very important things and they are very highly regulated. We have very regulatory bodies to get the approvals. Next slide, please. So some more important concepts. I know you are tired. Okay, right? So no more concepts. Okay. What's the time, Murthy? Still, do we have the time? We have 10 more minutes. Yeah. Can I close? Okay, right. Yeah. Thank you. So it's our privilege that Professor Dr. VVR sir came here. So I don't want to take any more time. So that's it. So we will be having this second show. This is a completely different concept, and we will have a talk on this because it's entirely a different thing. Thank you. Thanks for the opportunity, and it's my privilege to address all of you. It's something routine break to all of us. Thank you. Thank you so much.
Sir, for your invaluable contribution to our gathering today. It has been an eye-opening message to the audience. On behalf of everyone, we extend our heartfelt gratitude to our keynote speakers of the session. We are glad to have Dr. Venkateshwarlu sir among us. We request Dr. M. Sudhakar sir to kindly present the token to our honored guest, Dr. G. Chandresh. I kindly request all the dignitaries to participate in the felicitation. I request Dr. Venkateshwarlu sir to come on to the dais.